Hi, hello everybody. This is your master teacher from Biomentor Academy. Welcome to second video of A+1 short series of class 10 chapter 1 life processes. So, previous class we studied about what is nutrition, what are the factors essential for nutrition, what is photosynthesis? What are the raw materials required, external and internal factors? Next, we learn about the mechanism and significance of photosynthesis. So, to prove that those external factors are very, very essential for us, today we are going to study about the experiments on photosynthesis. Our first activity is to prove that the starch is present in the leaf. For that, we have to take a potted plant which must be well ventilated and where the sunlight will be fall. The plant must be exposed to the sunlight well. Such potted plant, the leaf we have to take out. Then, after plucking the leaf from the potted plant, we have to dip the leaf in a beaker containing the water. And we have to boil the leaf with the help of Bunsen burner. Why we need to boil the leaf to do this experiment? Because the leaf is a living. So, the enzymatic reactions will be taking place. To stop that enzymatic reactions, we need to boil the leaf in a water. After boiling the leaf for around 2 to 3 minutes, we have to take out the leaf and keep in a test tube containing methylated spirit. Means we can also say it is a type of alcohol. And that test tube, as you are observing in the figure, we have to keep in a water bath and we have to boil it. Why it is necessary to keep the test tube in the water bath? Because the methylated spirit is inflammable. It has an inflammable nature. Here we need to boil the leaf in the alcohol, that is methylated spirit. What is the need of boiling the leaf in the methylated spirit or the alcohol? Because to remove the chlorophyll pigments from the leaf, it is very, very essential to boil the leaf in the methylated spirit. For that purpose, we are keeping the test tube which contains this methylated spirit along with the leaf in a water bath. We have to boil it for 2 to 3 minutes till all the chlorophyll pigments are present in the leaf should be come out. Next, we have to we can see, we boil the leaf in a methylated spirit. Now all the chlorophyll pigments are come out. Now the leaf is pale in color. This leaf we have to wash with the water. After washing the water to become lukewarm water we have to wash. So the leaf must be soft. Next we have to pour the dilute iodine solution on the leaf. When you pour the dilute iodine solution on the leaf turns to blue-black in color. It seems that the starch is present in the leaf. This is the conclusion for this is the change of the color indicates that the presence of starch. In this way, we can prove that the leaf is preparing the food in the form of starch. So, our next activity is around chlorophyll is present. The green color part of the leaf contain the chloroplast which contain chlorophyll pigments which is helpful for photosynthesis. For that we have to take two types of plants. First one is variegated leaf means cotton leaves. You might have observed in this leaf the edges of the leaf is white in color and middle it is dark green in color. The part where the dark green in color is there, it contains more chloroplast which has more chlorophyll pigments so that it can able to prepare the food. And another plant, this is, you might have observed the leaf, the edges of the leaf has dark green in color, whereas in middle it is a pale yellow in color. It means the most of the chloroplast contained in the edges of the leaf. So, and next we have to take to prove that uh, normal leaf also we have to take it. Now what we have to do by taking this potted plant, maybe a variegated leaf or the normal potted plant, we have to keep the plant for 2 days in the dark. 2 to 3 days we have to keep in the dark. What is the need to keep the potted plant in the dark? To de-starch. So whatever the starch is present in the leaf. In the previous, we learned that the leaf contained the starch. So that if we will keep in the dark, the plant is unable to prepare the food. So whatever the starch present in the leaf, they will utilize that starch. It become de-starch. Then by taking that type of plant, we have to keep in the sunlight. After 2 to 3 days, the de-starched plant, what we have to do? Keep in a sunlight. When we kept in a sunlight for 6 hours, the plant is started to do the photosynthesis. Now we have to pluck, after 6 hours, we have to pluck the leaf from the potted plant and do the starch test. When we will do the starch test, the part you might have seen here, first I said, in middle of the leaf contain the chloroplast. That's why it is dark in color and sides are white in color. So this is the same leaf. You might have observed the white part of the leaf does not contain any chlorophyll. That's why it is brown in color. Whereas after boiling, it is the middle part is become green in color. And when this leaf, after boiling it, it becomes pale in color. And when we do the starch test, the diluted iodine solution when we pour, you might have observed that wherever the chloroplast is there, that part changes to blue-black and the remaining side edges of the leaf become brown. Means no chloroplast, wherever there is no chloroplast, no starch is prepared. So for this, we came to know that the chlorophyll, the chloroplast which contain the chlorophyll is very, very essential for the process of photosynthesis. In this leaf, you might have observed the middle, wherever the yellow color, no photosynthesis is taking place, no starch is prepared here. Wherever the green part, wherever the dark green is there, they contain the chloroplast having a chlorophyll pigments turns to blue-black color in the starch test. So, for this, it is proved that chlorophyll is very, very essential for photosynthesis. The next experiment, another external factor for us is the carbon dioxide. This carbon dioxide is very, very essential for photosynthesis. How can we prove that the carbon dioxide is essential for photosynthesis? For that, what we have to do, we have to take a potted plant when we are choosing the potted plant, we should choose such a potted plant which have a long and narrow leaves. Such type of potted plant we have to take. And this plant again we have to keep in a dark for 2 to 3 days to discharge the starch part. Discharge the starch part. Then we have to bring the potted plant and one of the leaf, as you can see in the figure, one of the leaf we have to insert in a conical flask or the round bottom flask which contain KOH solution. So, you might have observed the half of the leaf is insert inside the glass bottle with the help of the splitted cork, and half of the leaf is outside. So this whole setup now we have to keep in the sunlight. Take care. Here what precautions we have to take? The leaf, whatever is inserted inside the bottle should not touch to the KOH solution. One thing. Second thing, the cork should be fitted tightly. Okay, and it is easy for us to take a long, narrow leaf. These precautions we have to take. Then we have to keep this setup for 6 hours in the sunlight. After 6 hours, we might have seen that as we know that the half of the leaf, whatever is present outside, is able to prepare its food because from the atmosphere it is getting the carbon dioxide, whereas the inserted part which is present inside the glass bottle unable to prepare the photosynthesis because of the KOH. This KOH, what it will do? It will absorb the carbon dioxide. It doesn't allow the inserted leaf part to prepare the food. Okay, now we have to pluck the leaf from the plant after 6 hours in the sunlight. Then we have to do the starch test. When you did the starch test, the result is what we observe here. We observe that the plant leaf, the leaf which is exposed to outside is able to prepare the food because it get a carbon dioxide, and the inserted leaf which is present inside the bottle, glass bottle, is unable to prepare its food because unable to prepare the starch. Why it is unable to prepare the starch food? Because the KOH solution is absorbed the complete carbon dioxide. It proves that the carbon dioxide is essential for photosynthesis. Children, I hope you understood. So, who made first this experiment? Mole. So, this experiment is called Mole's half-leaf experiment. Next, the last essential factor for photosynthesis is sunlight, because the sunlight is only the source that the plant get energy. That energy, as we learn in the mechanism, that energy is helpful to absorb the light in the form of energy for the splitting of the water and chemical reaction. Light energy is converted into chemical energy inside the leaf. For that, the leaf has to trap the solar energy in the form of photon. The light, whatever the light energy the leaf is absorbing is called what? Photon. And now what we have to do to prove that the light is essential for photosynthesis. For that, as usual, we have to take this potted plant, keep in a dark for 2 to 3 hours to discharge the leaf. Next, what we have to do, we have to take one black paper as we have observed here, and we have to insert on the leaf, some part of the leaf, we have to cover with this black paper. Then next what we have to do, after doing this setup, we have to keep in the sun so that this potted plant is able to prepare its food. But wherever this black paper is inserted on the leaf, covered the leaf, it is unable to get the sunlight. So, it cannot prepare the food. So, for that, we will observe. You can see that again the starch. See here, we, with the help of these clips, we kept this black paper, and that black paper, when we have to keep, when the plant is, the leaf is attached to the plant, only we have to keep. After keeping this black paper, inserting the black paper with the help of the clips, then only the setup should be kept in the sunlight. After 6 hours, we need to pluck the leaf and we have to do the starch test. When we did the starch test, we will observe that the part where the black paper is clipped in, it is become colorless. And the remaining part, whatever is exposed to the sun, is converted into blue black. It shows that the sunlight is very, very essential for photosynthesis. While doing this experiment, what precaution should be taken? The precaution should be taken that the clips should be kept properly and tightly should be kept, and we have to take care that there is wherever the part which is covered with the black paper should not, sunlight should not enter through it in such a way that we have to keep the clips very tightly. So these are the factors which are essential for photosynthesis. So children, I think you understood this one, and these are the main experiments around in our 10th class examinations. Mostly sunlight is essential for photosynthesis and carbon dioxide is essential for photosynthesis. These are very important questions. So go through it for your examination. Next, why the iodine solution is used to test the starch, because it is the indicator. It will turns the wherever the starch into, wherever it prepared the starch into blue black as an indicator we are using. Why we are using the methylated spirit means what we will say, to remove the chlorophyll pigments we are using the methylated spirit. These are the main questions whatever we observe in this experiment, mostly common questions in the examination. So please watch this video. Any doubts you can keep the comments, and moreover our next video is about the heterotrophic mode of nutrition. Thank you.